Click for IB Biology. We're looking at the origins of cells. There are currently at least two major competing hypotheses, the hydrothermal vent hypothesis and the hot spring cycling hypothesis. So we'll take a look at those. The earliest cells are the progenotes. We'll take a look at the concept of that, and we'll consider the importance of the membrane and genetics in the evolution of the progenotes and our last universally common ancestor, LUCA. There's a PDF of this presentation down in the description, and there's a quiz that you can take to check your understanding. So let's take a look at the hydrothermal vent hypothesis. So in about 1977, this deep sea submarine was surveying in this region here, near the Galapagos, when it encountered what are now called hydrothermal vents. So these structures here are found at coincidences of the Earth's crusts coming together like this, and the volcanic activity that occurs beneath them, resulting in the production of very hot water at great depth. Now, the influence of this hot water is to change the solubility of minerals in the water, and to allow a form of what is called chemosynthesis. So this is a way in which biological molecules can be synthesized without the presence of light, and instead the organisms involved in these hydrothermal vents are drawing on the hot water from the volcanic activity, and instead of using water itself, they will be using hydrogen sulfide as a source of hydrogen for the reduction and fixation of carbon dioxide. The vents tend to be at depth, as we previously mentioned. They are hollow structures. They have food webs and food chains which are entirely contained within this structural limit. Outside here in the water is still dark, it's still cold with higher pressures, but at the location of the hydrothermal vents we have these very specific conditions which allow chemosynthesis. And so it was wondered whether or not this type of structure might be applicable to the Hadean period of time, about 4.5 to 3.5 billion years ago, in which the simplest of cells began to evolve. Was it actually under the similar conditions that we find in hydrothermal vents? And so the study of hydrothermal vents has been linked to the origin of cells and one hypothesis where life may have begun. This now has to compete with theories developed in the 21st century, in which it's claimed in what's called the hot spring hypothesis, more of a terrestrial rather than an oceanic argument, in which pools of hot water, which of course still exist in geologically active regions, create cycles of hot and dry, wet and dry rather, which allows the formation of polymer molecules within these rich, mineral-rich pools, and the formation of polymers has been shown to occur in these particular areas. The SETI Institute is responsible for a video, 1 hour and 15 minutes, I believe, which goes into the competing hypothesis in great detail, and I've left a link in the description so that you can watch that when you have time to do so. So two competing hypotheses about the possible location for the evolution of polymers and the simplest of cells. Those very, very early simple cells are known as progenotes. They are expected to have a very simple membrane structure, and there would also need to be a simple genetic structure, which is has been proposed as being based on RNA rather than the DNA with which we associate. We'll take a look in more detail at that in a moment. The conditions, of course, were anoxic, yet to become oxygen atmosphere. No oxygen in the atmosphere at that time, so we very much had a reducing environment conducive to building polymers, of course. And this lasted up till about 3.5 billion years ago with the archaea. And then at this time, the fossil record shows that we already have cyanobacteria, which were starting to carry out photosynthesis and create the oxygen atmosphere. So the early conditions would be anaerobic, and the later conditions started to move towards aerobic conditions. The progenome is a very, very simple structure. Life today has a common genetic code, as you will know, based on A, G, T, C, and U, the bases in the nucleic acids, and very, very similar genetics and protein synthesis, showing a commonality, a universality of the genetic code in all living things today. And in fact, this does allow comparisons between organisms using what is known as a molecular genetic clock, and this will be developed when we're doing in the genetics section of the course. All of this points back through time to somewhere past the 3.5 billion years ago, to a transition from this very simple progenome to what we would call LUCA, the last universally common ancestor. And this would be, here's LUCA, which would then begin to, in evolutionary time anyway, diverge into different types of cells and the diversity of life as we know it today. But the last common ancestor we had was called LUCA. Now LUCA would already have solved the problem of the boundary, which today we call the cell membrane, between the biochemistry of life and the abiotic world on the outside. This is what the membrane does. And the membrane controls what goes into and out of the cells. And we know today that this is composed of bilayers of what are called phospholipids. And the course actually goes into this topic in considerable detail if you look ahead to membrane structure for those details. These phospholipids, for the moment, we can describe them as creating these containers, these very simple cells in which the external environment is here and the internal environment is here, isolated from the external world. And the membrane, which forms this double layer called a bilayer, isolates it from the external world, but in turn also includes mechanisms of exchange between the external abiotic world and the internal biotic world. How this membrane structure evolved is also the subject of competing hypotheses, including one which is known as the clay hypothesis. And then, of course, as I've previously mentioned, there's also the hot spring theory. Within this early LUCA structure, it's postulated that the genetic material is RNA, ribonucleic acids, not the DNA of today. RNA has a number of features which make it a strong contender for the early genetic carrier, which include its capacity to code for structures, including the proteins, that it has enzymatic capacity, and also that it has the capacity for replication. Three factors important in these simple early cells. The role of RNA over the more contemporary DNA studies which have taken place has been highlighted in recent 21st century discoveries that there are more forms of RNA in our cells operating than proteins, which of course make our proteome. And the significance of RNA and RNA studies has highlighted that this very much is the strongest contender for the earliest genetic material. We're now going to go on and consider cell structure, and a link to the next video is appearing here with the next playlist highlighted and the subscription button should you wish to subscribe.